Any questions? Go ahead.
Could you talk us through sort of how does, as you're researching a question, how do you progress, or how do you progress from one of these methodologies to another? Are you doing them on the same subject at the same time? I'm guessing you can't wear the electrodes in the fMRI.
You can.
You can?
Yes.
So could you walk us
Yes.
through sort of, do you do the same things on the same subject? Do you do one thing and then get confirmational data that helps you just tease out causality?
Yep.
I know that this probably ties in well to the death of the fMRI.
Mm-hmm.
It's the awesome thing by itself, too. So.
Yep. Yeah, I mean, it's a good question. I really can only speak from my experience, and I don't know if it happens the same way for everybody, but my background is really in EEG research. So I did this as an undergraduate. I went on to graduate school in a lab that only did EEG research. And it's still sort of my passion, and that's what I'm doing at Bowdoin. But there's limitations of that method. So at a certain point, I got all this EEG evidence, but I said, really, to make the claims that I'm making, I want to know where in the brain that's happening, and I can't tell that from EEG. So what do? Well, I put the person in a scanner, and I give them the same task in a scanner. But even better, and this is what I did as in my postdoc, I did a simultaneous EEG and MRI study. So I took, I had all this evidence from EEG, and then I used that same paradigm and put people in the scanner, looked at the EEG, and then correlated the EEG activity with where it's happening in the brain from the fMRI. So it can be done. It's complicated, and there's,
And that still doesn't really sort of seal the deal with causality necessarily.
No, it's still correlational.
Things like transcranial stimulation to cause deficit.
Yep. Well, in the case for the research I want to do, it's not causing deficit, it's actually enhancing activity, but it can be done in either direction. But yeah, but you're right. All of these neuroimaging methods that I talked about are correlational. All I'm doing is giving people a task, and I'm saying, does that correlate with some activity that's happening in the brain? I mean, it's not causal, but it's pretty convincing if you have many, many studies that show the same activity pattern. But to truly say that area of the brain is necessary for this function, what I would have to do is either externally stimulate and increase the activity, or decrease the activity and show that it affects behavior.
Okay.
Go ahead.
I was wondering how atomized the field of cognitive neuroscience is, and how much literature you read outside of your field that might have something to do with your work. It just seems that there's so much being published, and it's becoming more and more specialized.
Yeah, I mean, it's kind of a tough question for cognitive neuroscience, because there is a field. There's a journal of cognitive neuroscience, but there really isn't. I mean, I read stuff from the journal Hippocampus down to journal of neuroscience. I mean, there's not really a series of journals that are only specific to cognitive neuroscience. There's one, but and there's a couple others that tend to be mostly cognitive neuroscience. But I think this is kind of why coming from my background, which was really in animal research as an undergraduate, I kind of liked cognitive neuroscience because it connects those levels. So I actually read probably anything from a neuroscience journal up to a cognitive science journal, which are slightly different ends of the spectrum of looking at animals and the molecules in the brain, up to these computer models of how cognitive functioning works. So, I mean, it's hard to say. I don't know. I don't go and look only for a journal that says cognitive neuroscience. I mean, I kind of just read anything that's related to the topic. Yeah, but I mean, that's a good point, too. I think there's also, which I didn't include in my future directions here, but I think in the field of cognitive neuroscience, I think it's also a bit frowned upon now if you only talk about animal research or you only talk about human research. They really want you to sort of combine across these different levels of understanding. So we want to know what's happening in the molecules in the brain up to the level of cognitive theory.
I have a question. You say your main interest is EEGs. Where do you get the EEG readings or data to do your research on it? Actually in clinical settings where you're...
No, it's here. So those machines, it's actually kind of nice. So these machines used to be huge and quite expensive. That's the size that my amplifier is about this big. I'm actually able to, if I wanted to take it to a different building on campus or take it to another institution or something, I can actually just move that. I've got a big suitcase I take with my EEG equipment in it. I mean, it's not quite that easy because you have to worry about electrical noise and stuff. Yeah. They're also better though so that's also, you used to have a Faraday cage, which is a completely metal enclosed structure that the person would go in to do. The amplifiers are better at filtering out noise now, so you can, it's fairly portable. So, but you don't have to do it in a clinical setting necessarily. The same thing with the MRI machine. Now, the MRI machine is really expensive. So this equipment is about $50,000 for one setup. The MRI machine is millions of dollars. So those you don't find as many at as many institutions, but they do have some that are dedicated for research. We have basically actually this exact same MRI machine that I showed you in this picture we have over at Maine Med, and some institutions will pay a clinical facility to use their scanner. So we would pay them, $500 an hour to do research on it. But other institutions have a research dedicated scanner, which is what we had at Brown, where I was for my postdoc. We had a scanner that was only for people doing research. They weren't bringing people in to do patient studies.
One last question.
Okay.
Do you work primarily with students, or do you work with population, people who have various disease difficulties, or where do you pull your information?
Yeah, so I primarily work with students. I have done one study at a hospital where I looked, so you can also get better activity from EEG if you record from inside the brain. So the people that go in for pre-op epilepsy will be recorded they'll have electrodes actually implanted within their brain and be recorded for like a week to see where the aberrant activity is coming from. And then sometimes they'll let researchers come in and do research with those patients. So I have done some of that research, but primarily I do research on normal, healthy young adults.